All right, so you guys, check this out. Scientists have actually gone and created the first ever, like, complete wiring diagram of a fly's brain.
It's pretty amazing stuff.
I know, right? It sounds kind of crazy when you first hear it, but this is huge. We're talking about potentially unlocking some major secrets about how brains work not just in flies, but in all sorts of creatures, maybe even us.
Yeah, it's true. This research is really about understanding these things called connectomes.
Connectomes.
Basically, picture a super detailed map that shows exactly how every neuron, like all those tiny little brain cells, are talking to each other in a network.
So kind of like a Google Maps for the brain, but zoomed in way, way closer. Wow, that's wild. But why a fly, though? Don't get me wrong, they're fascinating and all, but what makes their brains so special for this kind of research?
Well, fruit flies, they've been kind of these unsung heroes of neuroscience for like over a century. They're sort of like the lab rats of the insect world.
Really? Never would have guessed that. Why them?
Well, their brains, while still super complex, they're a lot more manageable to study than, let's say, a human brain. We're talking about like 100,000 neurons in a fly versus the billions and billions we've got.
Okay, yeah, that makes sense. Still, it's hard to imagine our brains having much in common with the flies.
Believe it or not, despite the size difference, fly brains actually share a surprising number of similarities with mammalian brains, including our own.
Whoa, seriously. So discoveries made in these tiny fly brains could actually tell us something about how our own brains work.
That's exactly the idea. And this new complete connectome, it's like taking a giant leap towards that goal.
Okay, I'm starting to see why this is such a big deal. But how did they even create this brain map? What kind of technology are we talking about here?
It's an incredibly meticulous process. It involves something called electron microscopy.
Electron microscopy.
Yeah, imagine taking a fly brain and slicing it incredibly thin. You were talking thinner than a human hair. Then they use these crazy powerful microscopes to capture images of each and every slice.
Wow, talk about painstaking. What happens next? They got to reassemble those images somehow, right? Like some kind of 3D puzzle.
Exactly. The researchers basically rebuilt the fly brain from those images, figuring out each individual neuron and then painstakingly mapping out every single connection between them. The sheer scale of this project is mind-boggling.
Okay, so just how many connections are we talking about in a single fly brain?
Get ready for this. This study mapped roughly 130,000 neurons.
130,000.
Okay. And a staggering 130 million synapses.
Hold on, synapses. Remind me what those are again.
Synapses, those are the tiny junctions where neurons transmit signals to each other.
Right, of course. 130 million. My mind is already buzzing just trying to picture that amount of data. Mapping it all is one thing, but how do scientists even begin to make sense of that kind of information?
Well, that's where FlyWire comes in. Think of it kind of like a 3D Google Maps, but for a fly's brain. Scientists can zoom in and out, explore all the different regions of the brain, and see how everything's connected at a level of detail we've never seen before.
This FlyWire thing, it's incredible. So what are researchers actually seeing when they zoom in on this brain map? Like, what's grabbing their attention?
Well, one of the most striking things is just how much of the fly brain is dedicated to processing visual information.
Yeah, it makes sense. I mean, have you ever looked at a fly's eyes? Those things are huge.
Right. We knew flies relied a lot on their vision,
Right.
but this really drives it home.
So it's not just about seeing, right? Like, what else can FlyWire tell us about how flies experience the world around them?
Well, it turns out flies aren't just seeing the world, they're experiencing this whole symphony of senses.
A symphony of senses, huh? I like that.
FlyWire, it's revealed all these connections between the visual parts of the brain and the areas that process smell, taste, touch, even sound.
Oh, wow. So it's like when we smell something delicious, let's say like freshly baked bread.
Yeah.
Suddenly it's not just the smell, right? You start picturing that golden crust, the warmth radiating from the oven, and then that anticipation for that first crispy bite. It's a whole sensory experience.
Exactly. That's a great analogy. And FlyWire, it's giving us this incredible glimpse into how all those different senses come together to create this one cohesive experience for the fly, shaping how it does everything. How it navigates, how it finds food, how it interacts with other flies, avoids danger, you name it.
You know, this is actually making me think about how we design things like robots and drones. Like maybe instead of focusing so much on vision for navigation, we should be thinking more about how to integrate all these other types of sensory information.
Exactly. FlyWire could be a goldmine for bio-inspiration in all sorts of technology.
It's pretty amazing, isn't it, how even these tiny creatures can inspire such groundbreaking innovation. Speaking of small, what about those 3 little eyes flies have on top of their heads? The ocelli. I always forget what they're called.
The ocelli, yeah.
What's the deal with those? FlyWire must have picked those up, right?
Oh, you bet. And those little ocelli, well, they turned out to be a lot more complex than we initially thought.
Really? How so?
See, scientists, we used to think they were just for basic light detection, like just telling the difference between light and dark, nothing too fancy.
Basic, right.
But FlyWire showed something way cooler.
Okay, I'm intrigued. What did they find?
Turns out, those ocelli, they're super densely connected to the parts of the brain that process visual motion from those big compound eyes.
No kidding. So they're not just sensing light, they're helping with motion.
Exactly. This suggests those ocelli are actively involved in things like helping the fly stabilize its gaze and coordinate its movements, especially when it's flying. It's like they have this whole intricate system for balance and orientation up in the air.
Wow, that's incredible. Those tiny little eyes are like mini gyroscopes. I'm telling you, we really underestimated the fly brain. But here's the question. What does all this tell us about our own brains?
Well, even though our brains are obviously way more complex, there are these fundamental similarities in how they're wired. And that's where the whole field of comparative connectomics comes into play.
Comparative connectomics. Okay, you're going to have to break that down for me.
Sure, think of it like comparing blueprints. Imagine you're looking at the blueprints for a bicycle and a car, right? They're both modes of transportation, but they're designed and built differently. By comparing them, we start to understand the basic principles of transportation, but also see how different designs lead to different capabilities.
Okay, I'm following you. So by comparing the FlyWire connectome to other connectomes, like, say, the roundworm one, which has already been mapped, we can figure out what's unique to each species and what's common to all brains.
Exactly. And the more of these connectomes we map, including the work that's being done right now on mapping a mouse brain, the more we'll understand about how brains have evolved and the basic principles of how they work. It's incredibly exciting stuff.
This is mind-blowing. But this brain map, it's not just a static picture, right? What else can scientists actually do with FlyWire?
Oh, this is where it gets really futuristic. We can use this super detailed map to actually create computer simulations of a fly brain.
Wait, brain simulation, like creating a virtual fly brain.
You got it. And by running these simulations, we can start testing out different hypotheses about how the brain works.
So, like, what kind of hypotheses? Give me an example.
For example, we can manipulate different connections in that virtual fly brain and see how it changes the fly's ability to do things, like learn new things, remember stuff, make decisions, things like that.
Okay, now my mind is officially blown. So we could potentially unlock secrets about how W learn and remember by studying a virtual fly brain. That's incredible. It's like we're peeking inside like a biological supercomputer or something. But these simulations, I mean, they could have applications way beyond just understanding how brains work, right? Could they actually help us build better technology?
Absolutely. Imagine using these simulations to like develop way more sophisticated AI or robots that can navigate these super complex environments.
But yeah, with that fly agility.
Exactly. Or even think about new therapies for neurological disorders. There's so much potential there.
This is seriously groundbreaking stuff. It feels like we're on the verge of like a total paradigm shift, not just in neuroscience, but everywhere.
And you know what's even more remarkable? The whole spirit of collaboration around all this. Like the FlyWire data, it's all open source.
Oh, wow. So any scientist from anywhere can just access it.
Yep. Scientists from all over the world can access it, contribute to it. It's amazing. A real testament to what we can achieve with collective brain power, you know?
It's like the whole scientific community coming together to unravel these mysteries. And all thanks to, well, the humble fly. Who knew those little guys were hiding such profound secrets?
It really makes you think twice the next time you see one buzzing around your fruit bowl, doesn't it? It's incredible, the intricate stuff happening inside that tiny brain shaping every single move.
It really does. I mean, it makes you realize that even the smallest creatures can hold the keys to unlocking these huge mysteries about, well, everything.
If a fly's brain can be this complex and this sophisticated, just imagine the potential locked away in our own minds. It's humbling, really, and incredibly inspiring at the same time.
Absolutely. It really leaves you with this sense of awe, you know, like a sense of wonder about, well, everything. And I'm so curious to see what other secrets these scientists will uncover next. And listeners, if you want to dive deeper into this whole FlyWire thing, you know, explore that incredible brain map for yourself. We'll make sure to link to the project in the show notes. You seriously don't want to miss it.
Definitely check it out. And be sure to stay tuned for more exciting updates from the world of connectomics. Like we said, this is only the beginning.
Until next time, stay curious, everyone, and never stop exploring.
Ever feel like your brain is buzzing with a million thoughts?
Well, get ready to have your mind blown because today we're taking a deep dive into a brain that literally buzzes the fly brain.
And before you click away thinking, really, a fly? Trust me, this is way cooler than it sounds.
We're diving into a study that's achieved something truly groundbreaking. They've mapped the entire neuronal wiring diagram of an adult fly brain.
That's every single neuron, every connection, a complete 3D roadmap of how information flows in this tiny but mighty organ.
So what are we hoping to uncover on this deep dive?
Today's mission is twofold. First, we're going to unpack why this achievement is such a big deal in the world of neuroscience.
And second, we're going to unearth some seriously fascinating details about the fly brain itself.
Think of it as a crash course in fly neuroanatomy, but with extra aha moments guaranteed.
To help us navigate this intricate landscape, we've got an expert on the line.
Let's dive in.
So you might be wondering, why all the fuss about a fly brain?
Well, it might surprise you to learn that the humble fly brain, despite being smaller than a poppy seed, is capable of some pretty complex behaviors.
These little guys can navigate, they can remember, they even engage in elaborate courtship rituals.
And here's the kicker. Many of the basic principles governing their brain function are believed to be similar to those found in more complex brains, including our own.
So it's like the fly brain is a simplified model system for understanding bigger brains.
Kind of like how engineers might study a miniature model bridge before building a full-scale one.
Exactly.
By decoding the fly brain, we're essentially getting a sneak peek into the fundamental principles of how brains in general process information, learn, and control behavior.
Now, this study didn't just take a quick glance at the fly brain.
They went full-on deep dive mapping out what scientists call the connectome.
Think of it like the ultimate wiring diagram.
And one of the things that jumped out at me was how much more complex this adult fly's brain is compared to its younger larval self.
It's a huge difference.
This study found that a whopping 3/4 of the adult fly's nervous system is packed into its brain.
That's a way higher proportion than in the larval stage.
And within the brain itself, a staggering 84% of the neurons are what we call intrinsic neurons.
Okay, pause for a quick definition check. Intrinsic neurons.
Remind us what those are again?
Absolutely.
Intrinsic neurons are the neurons that basically talk only to other neurons within the brain.
They're handling the internal chatter, so to speak.
This contrasts with sensory neurons, which are bringing information in from the outside world, things like sights, sounds, smells.
So a high proportion of intrinsic neurons means the adult fly brain is doing a lot of internal processing and problem solving, right?
It's not just reacting to the outside world, it's thinking things through.
Precisely.
It suggests a remarkable level of sophistication in how the fly processes information and makes decisions, especially compared to its larval stage.
Which makes sense, right?
A grown-up fly has got to deal with all the complexities of adulting in the fly world.
Exactly.
Think about it: finding food, avoiding getting swatted, engaging in those elaborate courtship rituals we mentioned earlier that take some serious brain power.
No kidding.
But before we get too carried away with fly love affairs, one of the things that really blew my mind was just how important vision is to these creatures.
It seems like a huge chunk of the fly's brain is dedicated to just seeing the world.
Oh, absolutely.
Flies are incredibly visual creatures.
They've got those amazing compound eyes that give them an almost 360-degree view of the world.
And then there are those three simpler eyes on top of their heads called ocelli.
They're thought to be involved in sensing light levels, which is crucial for things like knowing which way is up and maintaining stability during flight.
So it's not just that they see more of the world than we do. Their brains are actually wired to process all that visual information in a really sophisticated way.
Exactly.
And the researchers found that this emphasis on vision is clearly reflected in the brain's structure.
A huge chunk of the neurons they mapped, over 70,000, were located in the optic lobes.
That's where all that visual information gets processed.
Wow, 70,000 neurons just for vision.
That's like dedicating an entire city to processing visual information.
Laughs.
It is a massive investment in visual processing power.
And what's even cooler is how organized these optic lobes are.
Just like in the mammalian retina, they found a columnar structure with different columns of neurons handling information from specific parts of the fly's visual field.
Okay, break that down for me, a columnar structure.
What does that even mean in the context of a fly brain?
Imagine you're looking at a mosaic.
Each little tile represents a tiny piece of your visual field.
Well, in the fly's optic lobe, there's a dedicated column of neurons responsible for processing information from each one of those tiles.
So it's like the fly's visual world is divided into these tiny processing units, each handled by its own team of specialists. And within each column, you have different types of neurons responsible for detecting different features like edges, motion, and color.
Think of it like a neural assembly line. Each station with its own specific job to do to build up a complete picture of the world.
So it's not just a random jumble of neurons in there. It's highly structured, specialized, and organized.
You got it.
And it gets even more interesting.
The researchers found that not all neurons in the optic lobe follow this neat tiling pattern.
Some, like what are called LP12 cells, exhibit a more irregular, almost jigsaw-like arrangement.
So it's like evolution got a bit playful with those LP12 cells breaking the mold of the typical tiling pattern.
What's the purpose of that?
That's a question future research will hopefully answer.
But their very existence highlights the incredible diversity and specialization of neurons within even a brain as seemingly simple as a fly's.
It's mind-blowing, isn't it?
But let's not forget that all this visual processing isn't happening in isolation.
The fly brain needs to combine information from its other senses, too, right?
Absolutely.
And that's where another fascinating part of the fly brain comes into play, the SEZ, or subesophageal zone.
Think of it like Grand Central Station, but for sensory information.
Grand Central Station, love it.
So all the different sensory streams, sight, smell, taste, touch, they're all coming together in this SEZ.
Precisely.
The SEZ sits at the base of the fly's brain and acts as a central hub integrating all that sensory data.
This is where the fly puts it all together to create a unified perception of the world.
And it's not just about sensing the world.
This region also plays a crucial role in how the fly responds to what it senses right.
You're absolutely right.
The SEZ doesn't just receive information, it sends out commands too.
It connects to the fly's muscles and endocrine system, basically controlling how the fly reacts to all those sensory inputs it's receiving.
So if the optic lobes are like the eyes of the fly brain, the SEZ is kind of like the command center, taking in intel and issuing orders.
That's a great way to put it.
And what's really cool is that this study, with its complete connectome, gives us an unprecedented look into this command center.
It's like we've got the blueprints to the control room, and now we can start to figure out which buttons do what.
It's like having a peek behind the curtain of the fly's world, seeing how all those senses come together to shape its reality.
But let's zoom back in on one particular sensory pathway that really caught my eye, the ocellular circuitry.
Yes.
There's three simple eyes on top of the fly's head.
It's easy to overlook them with those massive compound eyes stealing the show.
Exactly.
But as we're learning, you can't judge a fly's eye by its cover right.
These ocelli might be simpler, but they're playing a vital role in how flies control their movements and navigate the world.
Absolutely.
And this study really dives deep into the ocellular circuitry.
They were able to trace the entire pathway from the moment light hits those ocelli all the way through the brain to the neurons that control the fly's wing muscles.
Talk about a full circuit analysis.
It's incredible.
So what did they find?
What are these ocelli actually telling the fly?
Well, remember we talked about how the ocelli are thought to sense light levels and help the fly figure out which way is up?
What they found is that neurons downstream from the ocelli receive a very specific pattern of input.
They get inhibitory signals from the eye on the same side of the body and excitatory signals from the eye on the opposite side.
Okay, I'm visualizing this.
Inhibitory signals from the same side, excitatory from the opposite.
So what happens if the fly starts to tilt, say, to the left?
You got it.
If the fly tilts left, the left ocellus will receive less light because it's shaded by the fly's body right.
This means less excitatory signals coming from the right ocellus.
At the same time, the right ocellus is more exposed to light, so it's sending out more inhibitory signals.
So it's like a seesaw, this balance between excitation and inhibition.
The fly tilts, the balance shifts, and the brain gets a signal that something is off kilter.
Exactly.
And that imbalance triggers a cascade of neural activity that ultimately makes its way down to the fly's wings, adjusting the wing movements to correct the course.
It's like a built-in gyroscope, helping the fly maintain stability during flight.
It's amazing how such a seemingly simple circuit can achieve something so complex.
And it highlights how crucial even these basic senses are for navigating the world.
Absolutely.
And it gets even cooler.
They found that this ocellular circuitry isn't operating in isolation.
It's actually talking to and receiving information from the compound eyes.
So it's not just a case of the ocelli controlling stability and the compound eyes handling everything else.
These two visual systems are working together, sharing information. They're integrated, creating a more robust and nuanced representation of the world.
It's like having multiple cameras with different lenses, each capturing a unique perspective.
You combine those perspectives and you get a much richer, more complete picture.
A perfect analogy.
And having this complete connectome allowed the researchers to uncover those intricate connections between the ocellular circuits and the visual processing centers in the brain.
It's like suddenly being able to see all the hidden wires connecting different parts of the brain, revealing how information flows through the entire system.
And speaking of information flow, that's another area where this study really shines.
Absolutely.
Beyond just mapping the physical connections, this research delves into how information actually travels through the fly brain, from sensory neurons all the way to motor neurons, with all those intricate stops along the way.
It's like they created a map of the fly brain's information highways, showing the main routes that signals take as they zip around the brain.
That's a great way to think about it.
They used a really cool approach called an information flow model.
This lets them estimate how likely a neuron is to be activated based on the strength and pattern of its connections to other neurons.
Okay, so it's not just about whether two neurons are connected. It's about how strong that connection is and how likely it is that a signal will actually travel down that path.
Exactly.
And what's really neat is that they could use this model to trace the flow of information from different starting points.
Let's say a fly smells something delicious.
They could simulate the activation of those olfactory sensory neurons in the fly's antenna, and then use the model to see where that signal travels in the brain.
So it's like dropping a virtual crumb on the map and watching as it gets carried along the information superhighway.
Exactly.
And what's really insightful is that they can do this for different senses, seeing which parts of the brain light up when the fly sees something, smells something, or feels something.
So what kind of insights did they gain from this information flow mapping?
Did they find any surprises?
Well, one thing they found was a really direct connection between the fly's sense of taste and its endocrine system, which is responsible for producing and regulating hormones.
So what, like the fly takes a bite of something and boom, its brain is telling its body to release a bunch of hormones.
Precisely.
And it makes sense, right?
Taste is a major factor in deciding what to eat.
And eating is going to have a direct impact on the fly's internal state, which is largely controlled by hormones.
It's like the fly's version of comfort food leads to happy hormones.
Exactly.
They also found strong connections between the fly's sense of touch and the motor neurons that control its legs and wings.
Again, a very logical connection.
Being able to quickly react to touch and pressure is crucial for things like avoiding predators or coordinating movement.
It's incredible to think that we can now trace these pathways in such detail.
It really paints a picture of how interconnected and elegant the fly brain is, even with its relatively small size.
It really is a testament to the power of this connectome and the analytical tools that researchers are developing.
But it's important to remember that it's still early days.
This information flow model is incredibly valuable, but it's still a simplification of what's actually happening in a living, buzzing brain.
Of course.
It's like having a detailed roadmap.
It shows you the main routes and highways, but it doesn't tell you about the traffic jams, the detours, all those dynamic real-world factors that influence how things actually move.
Exactly.
But even with those limitations, this research provides a fascinating glimpse into the functional organization of the fly brain and how those different senses are integrated to drive its behavior.
It really makes you appreciate the elegance of evolution, doesn't it?
Even a brain that's smaller than a pinhead can be this intricate and powerful.
But let's zoom out for a moment and talk about the bigger picture.
Because this study isn't just about flies, right?
It has implications for understanding brains in general, including our own.
Absolutely.
In fact, one of the most exciting aspects of this study is its potential for what we call comparative connectomics.
Now that we have these detailed wiring diagrams for both C. elegans, that tiny worm we talked about earlier, and the fly researchers can start comparing them side by side.
It's like holding up two blueprints and looking for the common architectural principles.
So we can start to see which features of brain organization are shared across different species, suggesting their fundamental principles.
And which ones are unique adaptations to a particular lifestyle.
Precisely.
It's like having a family tree, but instead of tracing genes, we're tracing the evolution of brains.
Seeing how they've adapted and specialized to different niches.
It's fascinating to think that the fly buzzing around your fruit bowl might hold clues to understanding the human brain.
It might sound crazy, but it's true.
And this comparative approach isn't just about satisfying our scientific curiosity.
It has real-world implications, particularly for understanding and treating brain disorders.
Because if we can identify those fundamental principles of brain organization that are conserved across species,
It stands to reason that what we learn from studying a fly brain could potentially translate to humans.
exactly.
And the fly brain, with its relatively small size and well-defined circuits, is a particularly powerful model for studying things like learning memory and decision-making.
We can manipulate those circuits in ways that are currently impossible in humans and see how it affects the fly's behavior.
So the humble fly could hold the key to unlocking some of the biggest mysteries of the human brain.
But let's talk about how they actually pulled off this incredible feat of mapping the entire fly-brain connectome.
Because it wasn't easy.
Not at all.
It was a monumental undertaking involving cutting-edge technology, sophisticated algorithms, and a whole lot of human brain power.
It all started with electron microscope images, right?
Like taking thousands upon thousands of ultra-thin slices of the fly brain and photographing each one.
Exactly.
And we're not talking about a few snapshots here.
They had to stitch together over 21 million images to create a 3D reconstruction of the entire brain.
It's like putting together a jigsaw puzzle where each piece is a microscopic slice of brain tissue.
Talk about a brain twister.
And then came the really challenging part.
Actually identifying and tracing individual neurons and their connections within that massive 3D image.
How do we even begin to do that?
Well, they used a combination of machine learning and good old-fashioned human effort.
The machine learning algorithms were used to initially segment the images, identifying different regions and structures within the brain.
So kind of like training a computer to recognize the different parts of a neuron, like the cell body, the axon, the dendrites.
Exactly.
But as sophisticated as these algorithms are, they're not perfect.
So a massive team of scientists and even citizen scientists from around the world painstakingly proofread and corrected the automated segmentation.
I read about that.
They created an online platform called FlyWire, where anyone could contribute to this massive brain mapping project.
It's amazing to think that people from all walks of life could play a role in such a groundbreaking scientific endeavor.
It was a truly collaborative effort, a testament to the power of open science and what can be achieved when people from diverse backgrounds come together to solve complex problems.
And it wasn't just about the technology and the manpower.
It also required a deep understanding of fly neuroanatomy.
Those scientists had to be able to look at a tiny segment of a neuron and know exactly what they were looking at, right?
Absolutely.
They had to differentiate between different types of neurons based on their shape, size, their connections. It requires an incredible level of expertise.
It's inspiring to see that level of dedication and passion.
And the best part is this incredible resource, this complete fly brain connectome.
It's now freely available to scientists all over the world.
Exactly.
And that's key.
This open access will undoubtedly fuel countless future discoveries.
Researchers can use this connectome to study specific circuits in detail, to test hypotheses about how the fly brain works, to develop new analytical tools and techniques.
The possibilities are really endless.
It's like they've given us a detailed map of a whole new continent, and now the real exploration can begin.
So what's next?
What are the next frontiers in connectomics?
Well, one obvious direction is to tackle even larger and more complex brains.
The human brain, with its billions of neurons and trillions of connections, is the ultimate prize.
Now that's what I call a deep dive.
But realistically, are we anywhere close to mapping a human connectome?
It's definitely a daunting task.
But this FlyBrain project has given us a roadmap.
It's shown us what's possible, and it's pushed the development of the technologies and analytical tools we'll need to take on that challenge.
So the FlyBrain is like a stepping stone, helping us build the tools and knowledge to eventually map our own minds.
That's incredible.
Precisely.
And who knows, maybe one day we'll be able to map the connectome of an individual human brain.
It's a bold vision, but imagine the possibilities.
We could have personalized neural atlases revealing the unique wiring patterns that make each of us who we are.
It's mind-blowing to think about.
We could gain unprecedented insights into how our brains develop, how they learn, how they age, how they're affected by disease.
Exactly.
This fly-brain connectome is just the beginning of a new era in neuroscience.
Well, I don't know about you, but my brain is definitely buzzing after that deep dive.
It's amazing to think that something as small and seemingly insignificant as a fly can teach us so much about the fundamental principles of how brains work.
It really makes you appreciate the incredible complexity and beauty hidden within even the smallest corners of the natural world.
Absolutely.
And on that note, we'll wrap up this deep dive into the fascinating world of the fly brain.
We hope you've enjoyed the journey as much as we have.
Until next time, keep exploring, keep questioning, and never underestimate the power of a tiny brain. Okay, so get ready, because today we're taking a look at something pretty incredible. We're talking about a complete wiring diagram of a whole adult fly brain.
Wow.
I know, right? It sounds crazy, but stick with me, because this is huge. Expert speaker, you're here to help break down just how big of a deal this FlyWire project really is.
Well, what's really fascinating here is we're seeing a major scientific breakthrough. I mean, this is the first time we've been able to map an entire adult brain's connectome.
And by connectome, you mean?
Right, the complete map of every single neuron and every connection between them. And not just for a fly, for any animal.
Wow.
It's a big deal. Up until this point, it's been like trying to get around with hand-drawn maps.
I like that analogy.
But this research is like finally getting Google Earth for the brain. We can zoom in on individual neurons, see all their connections in crazy detail. It's amazing.
So just to be clear, we're talking about the common fruit fly, right? Like the ones buzzing around your fruit bowl. Not exactly known for being the Einsteins of the insect world.
And yet, even with those tiny brains, they're way more complex than most people would think about.
Really?
Oh, yeah. Think about it. These little creatures can navigate, learn, remember, they can even fly in these crazy patterns.
That's true. I guess I never really thought about it like that.
All of that takes some serious brain power.
Okay, so how much brain power are we actually talking about here? Give us the numbers.
Okay, so this study, they found over 100,000 neurons and a mind-blowing 100 million synapses, and all of that in a single fly brain.
Wow, that's a lot to process for such a tiny creature.
It really is.
So what were some of the big surprises that came out of this brain map? What were the researchers like, whoa, didn't expect that?
Well, one of the major takeaways was how centralized the fly brain is.
Centralized?
Yeah, most of the neurons, they're mainly talking to each other, not directly to the rest of the body. It's like having a powerful central processing unit that's taking in information before it sends out commands.
So it's not just a bunch of separate circuits reacting to things around it. There's a whole lot of internal communication going on.
Fascinating. And it gets even more interesting because a huge chunk of the fly brain is dedicated to processing visual information, specifically the optic lobes.
Hold on a sec. We're talking about fruit flies here, right? They're not exactly known for having incredible eyesight.
And yet their visual processing is a lot more sophisticated than we thought. This suggests that vision is super important for their everyday life, more than we realized.
So they mapped out the entire structure of the brain, but they didn't stop there, did they? They actually tracked how information moves through the brain.
Exactly. They use a mathematical model to simulate how information from different senses like sight and smell could travel through that network in the brain.
So they're basically creating a simulation of the fly brain.
In a way, yeah. It let them pinpoint which areas of the brain might be working together to control specific behaviors.
Okay, I need an example. What's one finding that really blew your mind?
One of the most interesting discoveries was the ocelli circuit.
But what ocelli?
The ocelli. They're basically these simple eyes on top of the fly's head that can sense changes in light levels. Kind of like a really basic horizon detector.
Okay, I think I get it.
It's a primitive way for them to understand their orientation in space.
So what's so special about these ocellis?
Well, the researchers were able to trace the entire neural pathway from the ocelli all the way to the neurons that control the fly's wings.
You're kidding.
It's like mapping out every single connection in the neural circuit that makes you duck when you see something flying at you, but in a fly.
Wow.
This level of detail was impossible before FlyWire. It's incredible.
So they can literally see the information go from the eyes to the wings.
You could say that By mapping the connections, they can follow how signals from the ocelli might eventually affect the fly's wing movements.
That's incredible.
It's a huge discovery that gives us a never-before-seen look at how sensory information gets turned into action.
So it's not just a straight line from the ocelli to the wings, right?
Yeah.
I mean, there's got to be more to it than that.
Yeah. You're absolutely right. It's not that simple. The ocelli circuit actually gets input from the compound eyes, too. So it's combining that basic light-level information with more detailed visual data about movement.
So it's like the fly is using both sets of eyes, the simple ones and the complex ones, to get a fuller picture of what's going on around them.
Exactly. And what's so cool is that this kind of integration is probably happening all over the brain. We just couldn't see it before because we didn't have the complete picture.
So FlyWire is like finding the missing piece of the puzzle.
I'd say it's more like finding the whole puzzle.
True. We're finally figuring out how all these different parts work together.
It's like we're starting to understand the language of the brain.
Okay, but let's zoom out for a sec. We've been talking about flies. But if we can figure out how their brains work, what does that mean for understanding bigger, more complex brains like ours?
That is the big question, isn't it? The fly brain might seem kind of simple compared to ours, but remember, it's still incredibly complex with all those neurons and connections. If we can understand how this simple brain works, it could give us clues about how all brains work, including our own.
It's like cracking the code for a basic computer program.
Yeah.
Once you understand the foundation, you can start to see how it scales up to more complex systems.
Exactly. And this research could have huge implications for understanding the human brain, especially when it comes to things like learning memory, maybe even neurological disorders.
It's pretty mind-blowing when you think about it.
It really is.
So how did they even begin to make sense of all this data? They mapped the whole brain, but what then?
Well, that's where the idea of neuron superclasses comes in.
Neuron superclasses. Sounds like something out of a comic book.
Haha, kind of. They're like the superheroes of the fly brain. The researchers divided the fly's neurons into 9 different superclasses based on where they are in the brain and what kind of information they process.
So it's like organizing a giant library. You need a system to categorize all the books, otherwise you'd never find anything.
Exactly. These superclasses help us understand how the work is divided up in the brain. You've got your sensory neurons that bring information in from the senses, your intrinsic neurons that connect different parts of the brain to each other, and your efferent neurons that carry commands from the brain out to the muscles and other parts of the body.
And within each superclass, there are even more specialized types of neurons, right?
Absolutely. For example, within the visual projection neurons, there are different cell types that are specialized for detecting motion color, even the polarization of skylight.
Wait a minute, flies can see polarized light.
Yeah, it's actually more common in the animal kingdom than you might think. Lots of insects, birds, even some fish can see polarized light.
Wow, I had no idea. So what's the benefit of being able to see polarized light?
Well, for flies, it's thought to help them navigate, especially on cloudy days when they can't use the sun to orient themselves.
That's incredible. So by understanding these different neuron types and how they're connected, we can start to figure out how the brain processes information.
Exactly, like putting together a huge jigsaw puzzle. Each neuron type is a different piece, and the connectome shows us how they all fit together.
This is blowing my mind. But it also makes you wonder, if they had to map every single neuron and connection, just how long did this project take?
You're right. It was a massive undertaking. It took a huge team of researchers and a lot of time. They used all sorts of advanced imaging techniques, complex algorithms, even got some help from citizen scientists to analyze and map all the data.
Citizen scientists?
Really?
You mean like regular people were involved in this groundbreaking research?
Yeah. The researchers actually made some of the data public and invited anyone who was interested to help out with tracing neurons in the images.
No way. That's so cool. It shows how important collaboration and open science are.
Absolutely.
Okay, we've talked a lot about the structure of the fly brain. What about its function? Can we actually use this map to understand how the fly behaves in the real world?
That's the ultimate goal, and researchers are already working on it. They're using computer models to simulate how information flows through the brain based on different sensory inputs.
So they're basically building a virtual fly brain.
You could say that. They want to see if they can use this map to predict how a fly will react in different situations.
That's wild.
And one of the coolest things they've done so far is to trace that complete pathway we talked about from the ocelli, those simple eyes, all the way to the neurons that control the fly's wing muscles. They mapped the whole circuit that controls that specific behavior, which is a huge step towards understanding how sensory information becomes action.
So if they shine a light on the fly's ocelli, they can predict exactly how its wings will move.
That's the idea. They haven't actually done that experiment yet, but now they have the blueprint for it. This map is a powerful tool for understanding how sensory input gets turned into motor commands.
This is starting to sound like something out of science fiction.
I know, right?
We're not just looking at individual neurons or parts of the brain anymore. We're seeing the whole system working together.
It's like listening to an entire orchestra instead of just one instrument.
Exactly. Okay, before we go any further, let's back up a bit. This connectome we keep talking about, are we talking about an exact copy of every single fly brain? Like, is every fly wired in the exact same way?
That's a great question. It really makes you wonder, like, are we all just wired a certain way? Is there room for individuality even in a tiny fly brain? That's what makes this even more interesting. While there's a lot of consistency in the basic wiring of fly brains, it's not a one-size-fits-all situation.
Okay, good, because I was starting to get a little freaked out.
There's definitely room for individual variation, and that's what makes each fly unique.
So it's not like every fly is just a tiny robot programmed to act exactly the same way.
Not at all. That's what's so cool about biology, right? There's always variation and adaptation. And with FlyWire, we now have the tools to study those individual differences.
So you're saying we could take this baseline map and then compare it to connectomes from different flies to see how they differ.
Exactly.
That's incredible. We could see how things like genetics, experiences, even their environment might affect the way their brains are wired.
It's a whole new way of looking at how individual differences come about, and not just in flies, but potentially in all animals.
It's mind-blowing when you really think about the implications for studying learning memory, even how neurological disorders might show up differently in different individuals.
Exactly.
Okay, so we've talked a lot about the potential of this research, but what about the challenges? What are some of the limitations we need to keep in mind?
Well, one of the main things to remember is that right now, this is a static map. It's a snapshot of the brain at a single point in time. It doesn't show us the brain in action, how these neurons are actually firing and communicating with each other in real time.
So it's like having a map of all the roads, but not knowing the traffic patterns or the speed limits?
Yeah. That's a great way to put it. To really understand how the brain works, we need to see the dynamics of neural activity, how information flows through those circuits as the fly experiences different things.
So we need to find a way to combine this structural information with functional data to see the brain in action.
Exactly. And the good news is that researchers are developing some amazing tools that can actually record neural activity in flies with incredible precision.
Wow, so we could actually see which neurons are firing in real time.
And the FlyWire map gives us the context for understanding those recordings. We can see where those active neurons are in the circuit and how their activity relates to specific behaviors.
It's like having a blueprint of a city and then being able to overlay it with real-time traffic cameras and GPS data.
Exactly. You get a much richer understanding of how everything works together.
This is incredible research, but it sounds like there's still a lot we don't know.
Of course. This is just the beginning. There's always more to explore. For example, they haven't completely mapped the connections of all the photoreceptor axons from the compound eyes yet.
So there are still mysteries to uncover.
Absolutely. But that's what makes this field so exciting. This project is a perfect example of how collaboration, new technology, and a lot of hard work can lead to huge discoveries that change how we understand the brain and ultimately ourselves.
This whole deep dive has been incredible. It really makes you think, if a fly brain with all its complexity can be mapped out like this, imagine what we might discover about the human brain in the future.
It's an exciting time to be involved in neuroscience. There's so much more to learn.
And we're just starting to scratch the surface. That's what's so amazing about all of this. It's like we're on the verge of a whole new understanding of how the brain works and what it really means to be alive.
Absolutely. It's a journey of discovery, and we're all along for the ride.
Well said. And on that note, that wraps up our deep dive into the world of the fly brain and the incredible FlyWire project. A huge thank you to expert speaker for joining us and sharing your expertise.
My pleasure. Thanks for having me.
Okay, so get this. We're diving into a topic that's going to totally blow your mind, I think.
Oh, I agree. This one's a big one.
We're talking about the brain.
The brain.
But not just any brain.
No, not just any brain.
This deep dive is all about the first ever complete wiring diagram of an adult fly brain. Like, are you kidding me?
It's incredible, right? A complete connectome, every neuron, every synapse, all mapped out.
I can't even imagine.
It's not just a snapshot of a brain region either. It's the entire adult fly brain laid bare.
And this is a huge deal, right? This paper, Neuronal Wiring Diagram of an Adult Brain, it involved a massive team of researchers just to make it happen, it seems like.
Yeah, tons of people involved. But, before we even get into all the details, it's important to remember why the fly brain? Why is that so important for understanding brains in general?
Right, because when you think about it, a fly's brain, it's tiny.
Tiny, but mighty.
It's amazing what they can do.
Exactly. Flight, navigation, complex social interactions, all from this tiny little brain.
So it seems kind of counterintuitive. Wouldn't you expect a simpler organism to have a less complicated brain?
It's true, their brains are small. But it's not really about size. It's about the complexity of the organization and the connections. And, flies share a common ancestor with our own brains way back in evolutionary history. So by studying their brains, we can actually learn a lot about the fundamental principles of how brains work in general.
Like the Rosetta Stone of brains, then.
Exactly. This paper, what's really cool is they were able to analyze brain-wide activity for things like sensory input and motor function in a way that we've never been able to do before.
So what kind of new things are we learning then? What was the big aha moment here?
Well, think about it this way. In the past, we could only really study small sections of the brain in isolation.
Kind of limiting, I would imagine.
Yeah, exactly. It's like trying to figure out a city's traffic flow just by looking at individual intersections.
You're never going to get the full picture that way.
Exactly. This connectome, what it does is it gives us the entire picture, the complete traffic flow of information in the fly brain. We can see how visual input gets translated into a course correction during flight, how the brain decides to prioritize smell oversight when it's searching for food. It's amazing.
So it's like we finally have Google Maps for the fly brain.
Exactly. We can zoom in on specific circuits, see how different brain regions are connected, and really start to understand how the whole system works together.
And one other thing that I found totally mind-blowing in this paper was the density of the connections in that tiny space.
It's pretty incredible. They found over 100,000 neurons and 50 million synapses packed into a space smaller than a sesame seed.
That's insane. How is that even possible?
That's the beauty of it, right? It just shows how incredibly efficient nature can be. That density, it's about 7 synapses per cubic micrometer, which is way higher than what we see in the human cortex.
Wait, really? Our brains are less dense than a fly's.
It seems counterintuitive, right? But the human cortex, it has less than 1 synapse per cubic micrometer. So the fly brain is packing a whole lot more connections into a much smaller space.
So it seems like size isn't everything when it comes to brains.
Clearly not. And that density, that's probably a big part of why flies are so incredibly fast. Think about it. They can process information and react so quickly, it's almost impossible to swat them.
Oh yeah, they're like little ninjas.
Right. It's amazing.
So we've got this densely packed network of neurons.
Yeah.
But how does it actually work? Can you walk us through the general layout of a fly brain? What are the main parts and what do they do?
Sure. So imagine a bustling city. The central brain, that's kind of like the downtown core where a lot of the big picture stuff happens. And then you have these 2 prominent structures on either side. Those are the optic lobes.
And those are all about vision, right?
You got it. They're dedicated to processing all that visual information, which makes sense, right? Flies rely heavily on their vision for survival. And then there's this area called the SEZ, the subesophageal zone, which is kind of like the city's central station.
Why a central station?
Well, it's the main hub for all the non-visual senses. Smell, taste, touch, they all come together in the SEZ. And it's also a major output center. This is where a lot of motor commands originate, so it controls things like movement.
So the SEZ is like the brain's connection to the outside world.
Exactly. It receives input from the senses and sends out commands to the muscles. It's the brain's way of interacting with the world.
So we've got these different neighborhoods, each with their own specialty. But how do they actually communicate with each other? The paper mentions these different types of neurons, right?
Right. Think of it like the city's transportation system. You've got afferent neurons. They're like the delivery trucks bringing sensory information into the brain from the rest of the body. Then you have efferent neurons, which are like those trucks heading back out, carrying motor commands and other outputs away from the brain. And then finally, you have intrinsic neurons.
What do those ones do?
They're like the city's internal network of roads and subways. They handle all the communication within the brain itself.
Fascinating. So most of the brain's chatter is internal. What are they even talking about among themselves?
That's the million-dollar question, right? And it turns out that the vast majority of neurons in the fly brain are actually intrinsic.
Wow, so most of the action is happening behind the scenes.
It seems that way. It really challenges our assumptions about what a simple brain is doing.
Definitely makes you wonder what's going on in those tiny little heads. But before we dive into the implications of all this internal chatter, let's talk about how the researchers actually created this connectome in the first place. Like, how do you even begin to map something as intricate as a brain? It sounds like a monumental task, to be honest.
Oh, it definitely is. It took a massive amount of work and some really advanced technology. They used this technique called serial section transmission electron microscopy. It's a mouthful, I know. STEM for short.
STEM, got it. But what does it actually do?
So basically, imagine slicing the fly brain into these ultra-thin sections, like thinner than a human hair.
Wow, that's thin.
Right. And then they take these incredibly high-resolution images of each section with an electron microscope.
So it's like creating a super detailed digital flipbook of the fly brain, slice by slice.
Exactly. And then the real magic happens. They use these sophisticated computer algorithms to analyze all those images, identify individual neurons, and trace their paths through the brain. It's like putting together a giant 3D jigsaw puzzle.
I bet. So it's all done by computers then?
Well, not entirely. They actually had help from real people, too. They used a platform called FlyWire to enlist citizen scientists from all over the world to help proofread and refine those reconstructions.
Wow, that's so cool. So it's a combination of cutting-edge technology and good old-fashioned human observation.
Exactly, because sometimes you just can't beat the human eye when it comes to spotting those subtle details. And it's amazing because it means that anyone can contribute to this kind of groundbreaking research.
Like a global brain mapping project.
Yeah,
This whole project is seriously making me rethink how we define simple brains.
I know, right? We tend to underestimate the processing power of organisms that we think are less complex than us. But this research shows that even a tiny fly has a brain capable of remarkable things.
Absolutely. Okay, so you mentioned earlier that the paper goes into some detail about the types of connections they found. And one thing that really stuck with me was the idea that not all connections are created equal.
Right. Not all connections are the same. Some are stronger than others.
So how do you even measure something like connection strength between two neurons? What does that
Yeah.
even mean?
It all comes down to the synapses, those tiny points of contact between neurons. The more synapses you have between two neurons, the stronger the signal transmission. Think of it like the difference between a dirt road and a multi-lane highway. The highway can handle a lot more traffic, right?
That's a great analogy. So a higher number of synapses means a stronger, more influential connection.
Exactly. It's like the difference between a whisper and a shout. And when the researchers looked at the connection strengths in the fly brain, they found some really interesting patterns. Some pathways, especially those involved in processing sensory information and triggering those really fast motor responses, like escaping from a SWAT, those pathways have significantly stronger connections.
So it's like the fly brain is prioritizing certain pathways over others, making sure that those essential signals get through quickly and efficiently.
Exactly. It's fascinating, isn't it? This connectome. It's giving us this incredible glimpse into how the brain prioritizes information and makes sure those crucial signals get the express treatment. It highlights those pathways that are absolutely critical for the fly's survival. And, this kind of insight, it's not just about flies. It could have implications for understanding how brains prioritize information in general.
That's amazing. But this connectome, it's basically a static map, right? It shows us the connections.
Right.
But how do we know how these circuits actually function in real life?
That's a great question. And that's actually one of the most exciting frontiers in connectomics right now, moving from these static maps to dynamic models. And this paper, it actually hints at that possibility using this connectome for brain simulation.
Wait, so they can actually create a virtual fly brain in a computer, like a simulation?
That's the idea. They can take this incredibly detailed wiring diagram and use it to create a computer model of the fly brain. Then they can run simulations, test different inputs, and see how the virtual brain responds.
That's incredible. It's like having a virtual laboratory for the brain. You could test how the brain reacts to different visual stimuli or how it coordinates its movements during flight. The possibilities seem endless.
Absolutely. It opens up so many doors for research. We can start to ask really specific questions about how different parts of the brain work together to generate behavior.
So it's not just about mapping the structure of the brain anymore. It's about understanding how that structure actually gives rise to behavior in the real world.
Exactly. And that's what makes this research so groundbreaking. It's pushing the boundaries of what we thought was possible.
This whole conversation has me thinking about the bigger picture now. How does this research on the fly brain connect to the larger field of neuroscience? What are the broader implications here for understanding brains in general, even our own?
That's the million-dollar question, right? And it's what makes this research so compelling. Because even though we're talking about flies, the insights we gain from this connectome could have implications for understanding brains across the board.
Because at a fundamental level, all brains share some basic principles of organization.
Exactly. And this is where the field of comparative connectomics comes in. By comparing the wiring diagrams of different species, we can start to see what features of brain organization are conserved across evolution and which ones are unique adaptations.
So, like, are there any other connectomes we can compare this fly brain to?
Actually, yeah. The paper briefly mentions C. elegans, a tiny worm that's been studied extensively. They have a complete connectome for C. elegans, too.
And are there any similarities between the fly and the worm connectomes?
You might be surprised to hear this, but yes, there are actually some striking similarities in their underlying organization. It's like finding common architectural principles in buildings that look completely different on the surface.
So even though they have vastly different lifestyles and behaviors, their brains might share some fundamental design principles.
Exactly. And as we build a library of connectomes from diverse species, from worms to flies to mice to humans, we can start to really unravel the evolutionary history of the brain and the fundamental principles that govern its function. It's incredibly exciting.
It's amazing to think we can compare wiring diagrams like they're blueprints, you know? And it wasn't that long ago, mapping even one neuron seemed impossible. What changed the game to get us to fly brain connectomes?
Honestly, a lot of it boils down to just how good electron microscopy's gotten. The detail, how fast they can image brain tissue now, it's mind-blowing down to the nanoscale. But even that's only half the battle.
Because then you've got to make sense of it all.
Exactly. We're talking petabytes of data, like 1,000,000 gigabytes. It's an insane amount of information to process, even for computers. Thank goodness for those, by the way. I was reading about some of the early connectome projects.
Oh, tell me about it. Painstaking doesn't even begin to describe it, right?
It was all done by hand, basically, neuron by neuron, tracing those connections. Amazing dedication, but so slow. But that's where all this new tech comes in, machine learning, AI. We can automate a huge chunk of that process now. Computers can identify and trace neurons through those massive data sets way faster and more accurately than we ever could.
And speaking of speeding things up, wasn't there something in this paper about citizen scientists?
Oh, yeah. They use this cool platform called FlyWire. Basically, they crowdsource the proofreading of the connectome. Anyone could go online and help check the computer's work, make sure those connections were traced correctly.
I love that. It's like a global brain mapping party. Everyone chipping in to understand these incredible organs. Speaking of which, I got to ask about the ocelli. We touched on them earlier, how they help with flight control. But the paper got me thinking. It's wild how we can trace the whole circuit now.
Oh, yeah. From those simple eyes all the way to the output, the behavior. In this case, it's the motor neurons controlling the fly's wings. So they mapped how the fly brain takes in that light information from the ocelli and turns it into commands to keep the fly flying straight, even when the light changes suddenly.
It's like we could see the thought process, but in terms of actual physical connections. Seeing is believing,
Yeah.
or maybe in this case, seeing the connections helps us understand.
Totally. And it's not just about flies, right? This is how brains work, period. Sensory input to behavioral output, all through these incredibly complex circuits. The fly brain's just a model, but the principles, they're likely universal.
Which makes me wonder, where do we even go from here? What's next for connectomics? Are we going to map a human brain anytime soon?
I mean, that's the dream, right? But baby steps. A human brain is crazy complex. But we're getting there, one step at a time. Right now, folks are focused on mapping specific regions in super high detail. Like there's a project trying to map the entire mouse visual cortex.
Breaking it down, tackling it piece by piece.
Exactly. And every time we map one of those pieces, we learn more about how the whole system fits together. Plus, the technology keeps getting better, faster, more powerful. And it's not just about connectomics and isolation anymore. People are combining it with other techniques, like transcriptomics.
Which is, remind me again.
Basically looking at which genes are turned on or off in different cells. So not just the wiring diagram, but the molecular nuts and bolts of how those circuits are built and how they actually work. It's getting really, really granular.
Like connectomics is going to revolutionize everything.
Yeah.
Not just how we think about the brain, but medicine, computers, heck, maybe even what it means to be human.
I'd say we're well on our way. And this little fly connectome, it's a perfect example. Sometimes the biggest discoveries come from the smallest creatures. It just goes to show you never know where that next breakthrough is going to come from.
This whole deep dive's been incredible, honestly. From these teeny tiny fly brains, we've gone all the way to the future of connectomics, the nature of intelligence, the whole shebang. I feel like my brain's gotten a serious workout just trying to wrap my head around it all.
Me too. It really makes you appreciate the power of curiosity, and the dedication of these researchers to spend years, decades even, teasing apart these mysteries. They're like explorers, charting the unknown territories of the brain.
Absolutely. And on that note, we'll leave you with this.
Hmm.
Next time you see a fly, don't swat it right away. Take a second to think about that incredible complexity buzzing around inside its tiny little head. You're looking at a miniature marvel of engineering, millions of years in the making. And who knows, it might just hold the key to unlocking some of the brain's greatest secrets. Thanks for joining us for this deep dive, and we'll catch you next time.
Okay, get this. Are you ready for this? We are diving deep, deep into the world of the fly brain today.
Well, when we say fly brain, we're really talking about the Drosophila melanogaster, the fruit fly. It's incredible, really. Scientists have actually created the first ever complete wiring diagram of an adult fruit fly brain.
Oh, wiring diagram. For a brain, like, are we talking circuits and pathways here?
Yeah, exactly like that. We are talking about 100,000 neurons, millions upon millions of connections, all mapped out. And we've got excerpts from the actual scientific paper, Neuronal Wiring Diagram of an Adult Brain, to guide us, along with some really cool behind-the-scenes information about this research.
Okay, so this paper, it sounds like a landmark achievement, but how do they even begin to map something as complex as a brain?
It's not about flies exactly. It's way bigger than that. This paper helps us understand how any brain processes information, especially one this complex.
So let's unpack that, shall we? How did they even map this thing? I know they used electron microscopes, but this must be on a whole other level, right?
Oh, absolutely. Picture this. They take the fly brain and they slice it incredibly thin, like thinner than a human hair.
Wow, and each slice goes under an electron microscope, right?
Yep, each one. They capture these super, super detailed images. It's amazing.
Okay, so they have thousands of images, millions even, of these tiny, tiny brain slices. How do they even begin to make sense of that? That's like the world's most difficult jigsaw puzzle.
That is where it gets really cool. That's where AI comes in.
AI, like artificial intelligence.
Yep. They use these highly sophisticated algorithms, really cutting-edge stuff, to put the image puzzle back together. These algorithms trace the tangled paths of all those neurons.
Hold on, back up a sec. AI actually reconstructing a brain from slices. That sounds like something straight out of a sci-fi movie.
It's truly remarkable, and it allows us to see the brain's connectome.
Connectome. Okay, gotta admit, that's a new one. So what is a connectome exactly?
The connectome is like a map of every single connection in the brain.
Wow, so it's like having the full roadmap of the fly's brain. We can see where every signal goes.
Yeah, exactly. Although I will say,
Yeah.
it wasn't always smooth sailing. Early on in the research, they accidentally flipped the brain image.
They flipped the brain. How do you even accidentally flip a brain, like left to right?
Left to right, yep. It sounds funnier than it was, I assure you. Thankfully, it was an easy fix, but a good reminder that even with all of this extremely advanced technology, that human element is still crucial.
I can imagine. So how did they catch that?
Well, they had a whole team of scientists and volunteers, nicknamed Flyers, proofreading the AI's work. Talk about dedication.
Wow, a human-AI partnership to map a brain. That's kind of reassuring, actually, in a weird way. But what did they actually find? What's so special about this fly brain?
Well, for one, it's incredibly centralized. 3/4 of the fly's entire nervous system is packed into its brain.
3/4. Wow. That's a lot of processing power for such a tiny creature.
Right. It makes sense, though, when you think about it. They need all that brain power to navigate, to fly, to find food, to do these really elaborate courtship dances, and they do it all with a brain the size of a poppy seed.
That is wild. And the researchers actually traced the circuits in the brain from where to where?
From their simple eyes, called ocelli, all the way to their motor neurons.
Okay, so we're talking about understanding how a fly sees and how that visual input becomes action. That's huge.
It is huge, and again, it goes way beyond flies. The techniques they used here, this mapping of the connectome, could be used to map even larger, more complex brains in the future.
You mean like eventually?
Or
Ours. Okay, now that is mind-blowing.
it is mind-blowing. Because if we can understand the connections in a fly brain, imagine what we might learn about our own brains. And we're just scratching the surface here. What's really remarkable is the level of detail in this connectome. I mean, we're talking about seeing individual connections between neurons here, right?
Precisely.
Yeah.
Like we can actually trace the path of a single signal through this network of billions of connections.
Wow. So what does that even look like?
Imagine zooming in on a Google map.
Okay.
But instead of streets and buildings, it's neurons and synapses. That's the level of detail we were talking about here.
Okay, that's a trip. So we have this incredible map, but what does it actually tell us? What new questions can we even ask now?
Well, for the first time, we can actually study how information flows through different parts of the brain.
How different regions interact, how sensory input actually translates to behavior.
Oh.
It's like having a backstage pass to the brain's control room.
Whoa. So can we see, for example, how the fly processes what it sees?
Exactly.
Does it use just those compound eyes, or do the simple eyes, the ocelli, play a part too?
That's the amazing thing. It's not just separate pathways for seeing, smelling, flying.
Right.
This connectome shows us that there's a ton of crosstalk between different brain regions.
So the fly brain is more like a network then, than a bunch of isolated departments.
Exactly.
Yeah.
And we are seeing these really fascinating interactions. Like, for example, visual input from those compound eyes.
Yeah.
It's combined with information from the ocelli, those simple eyes on top of the head.
So the fly uses both types of eyes together to get a more complete picture of the world.
It seems that way. And it gets even more intriguing. There are connections between the visual system and their other senses, like their sense of smell.
Hold on. Sight and smell are connected. Does that mean a fly can, like, see a smell?
Not exactly. But it suggests that there's a level of sensory integration that we didn't fully grasp before.
Wow.
The fly's brain is processing information from all these different senses in ways we're just beginning to understand.
So this tiny fly brain, it's not so simple after all, huh? This connectome is really showing us just how complex their sensory experience might be.
Absolutely.
And it makes you wonder, what about us? We often think of our senses as separate.
Yeah.
Sight, smell, hearing.
Right.
But what if our brains are integrating them in ways that we don't even realize?
It's a fascinating question, isn't it? And it's something that we can start to explore.
Okay, now that is a mind-blowing thought. So this research on flies, it could actually change how we think about how our own brains work.
This fly-brain connectome is giving us this whole new framework for understanding brains in general. It's really pushing the boundaries of neuroscience. It's opening up so many avenues for research.
It's like we have stepped into a whole new world, and it is all thanks to this tiny fly brain. But what is next? Where do we even go from here? We've got this incredible map. Now what?
Now we dive deeper. We can start to pick apart specific circuits within the fly brain.
Okay.
Try to understand how they control those really complex behaviors that we talked about. Flying, feeding, courtship.
So we have got the blueprint.
Exactly.
And now it's time to figure out what each part does, how they all work together.
And it goes way beyond flies.
Yes.
Right? Like you mentioned, this research, it can be applied to other organisms too.
Right. So we could one day have a connectome of a mouse brain, or even a human brain.
It's definitely a possibility. The challenges are huge, obviously. Like these brains are exponentially more complex.
Yeah.
But the potential payoff, it is enormous.
Imagine having a complete wiring diagram of the human brain. That's like the next frontier of neuroscience.
It really is. And it's not just about mapping. Once we have these connectomes,
Yeah.
we can start building computational models, testing how they work, simulating different conditions.
Okay, so we could like simulate a brain injury.
Yes.
Or a disease and see how the connectome changes.
Exactly. Or test out new drugs and therapies.
Wow.
See how they affect the brain circuits.
That's incredible.
The possibilities, they are truly mind-boggling.
It really is. This is incredible. This tiny fly brain has opened up a whole new world of possibilities for neuroscience.
And it's important to remember that this is just the beginning.
Yeah.
The field of connectomics is moving so fast, and who knows what breakthroughs are just around the corner.
Well, I am hooked. This fly brain research has really captured my imagination, but we've talked a lot about the connectome itself, the possibilities. What about the how? How did they actually capture these incredibly detailed images of the fly brain in the first place?
That's a great question. And it takes us into the world of some seriously advanced technology.
We are talking electron microscopy here, but I'm guessing it's not the kind you see in a high school biology lab, right?
You got it. This is like next level stuff. It's called serial section transmission electron microscopy, or STEM for short.
STEM. Okay. That sounds pretty intense. What is that exactly?
Well, remember that bread analogy?
Yeah.
Slicing the fly brain super thin.
Yeah.
That's what they do with STEM. Each slice, each one goes under this really powerful electron microscope. And it uses a beam of electrons instead of light to create these really, really detailed images.
So they end up with thousands, millions even, of these microscopic images. How on earth do you piece them all back together to get a 3D view?
You guessed it, AI to the rescue again. They use these really sophisticated algorithms to align all those images. They stitch them together seamlessly. It's like solving a massive, massive 3D jigsaw puzzle, but on a computer.
That just sounds like a computational nightmare, honestly.
It's a huge challenge, for sure, but advances in computing power and AI, well, they've made it possible.
Okay.
And the tech is always improving, right? It's getting faster and more precise all the time.
So what's next for electron microscopy? Are there even more powerful tools on the horizon?
Oh, absolutely. Scientists are always pushing those limits. New electron microscopy technologies are emerging all the time.
Okay.
They promise even higher resolution, even faster imaging speeds.
So higher resolution, that means we'll be able to see even more detail in the connectome. Like what?
Exactly. Imagine being able to trace the connections between neurons with even greater precision. Even seeing the individual synapses themselves. That's the level of detail we're talking about here.
Wow, a microscopic look at the brain's wiring. Incredible. And then faster imaging. That means we can map larger brains more quickly, right?
All right, so it's not just about flies anymore. This tech could unlock the secrets of all brains,
Wow.
from the simplest to the most complex. It's a really exciting time to be studying the brain, that's for sure.
It really, really is. This whole deep dive has been incredible. We've learned so much about this groundbreaking research, from the tiniest fly brain to the future of neuroscience. It's amazing.
And, it really highlights the importance of scientific curiosity. Following that trail of discovery wherever it may lead.
Yeah.
Sometimes the most profound insights come from the most unexpected places.
That's so true. Who knew that a tiny fly could hold such huge secrets? Thanks for taking this deep dive with me. It's definitely left me with a lot to think about.
The pleasure was all mine, truly. And for you listening out there, keep exploring, keep asking questions. Who knows what other mysteries are waiting to be uncovered?